So now let us take these questions. The first question is, by looking at a plant externally, can you tell whether a plant is C3 or C4? Why and how? So in this question they want to find out, can we differentiate between a C3 plant and a C4 plant from outside, externally? If we see two plants, can we just say that this is C3 and this is C4? Our answer is no. Externally, C3 and C4 plants cannot be distinguished. Because morphologically they are identical. Yes, they can be identified if we go to the inner structure, that is anatomical. So we can differentiate by studying its anatomy. Like we talked about C4 plant showing Cran's anatomy, the bundle sheath. So if you see that, it is easy to tell whether it is a C3 plant or a C4 plant. But from external appearance, we cannot say, because morphologically they are identical. Second question. By looking at which internal structure of a plant can you tell whether a plant is C3 or C4? Just now we were talking about this. So by looking at which internal structure. So if bundle sheath, if internal structure, that is bundle sheath, which is wreath-like, bundle sheath, then we can say it is C4. If there is no regular arrangement of the cells around the vascular tissue, then we can say it is C3 plant. We can also distinguish between these two on the basis of two types of chloroplast. Dimorphic, so this is one point. Second, if we see dimorphic chloroplast, then we can conclude that the plant is C4 plant. So internal structures can help us distinguish between C3 and C4. Third question. Third question is, even though a very few cells in a C4 plant carry out the biosynthetic Kelvin pathway, yet they are highly productive. Can you discuss why? They are saying, the question says that there are few cells in C4 plants where Kelvin cycle takes place. Let us talk about C3 and C4 to understand this question. In C3, the upper epidermis, the lower epidermis, here is xylem and phloem, and the cells which are here, all these cells, the spongy parenchyma or palisade parenchyma, they all have chloroplast and they perform photosynthesis, that is Kelvin cycle. So where is Kelvin cycle taking place? That is C3 cycle takes place in all mesophyll cells. Whereas in case of C4 plants, C3 cycle takes place only in bundle sheath. So C3 cycle is only in bundle sheath cells. So now they are asking that even though very few cells of C4 plants carry out this Kelvin cycle, yet their productivity is higher. Why have they undergone this adaptation? This adaptation in C4 plants has taken place to avoid photorespiration. So, in case of C3 plants, carbon which is fixed by photosynthesis is, or let us write carbon fixed by photosynthesis is lost by photorespiration. That is why productivity. What is meant by productivity? Productivity is using the carbon, how much of organic matter is synthesized. So, photosynthesis fixes this carbon and photorespiration releases this carbon. So, the end result is less. Suppose 100 carbons are fixed and 25 are lost by photorespiration, only 75 remain. But here there is no loss of carbon by photorespiration. So, here there is no loss of carbon because there is no photorespiration which is seen in C4 plants, and that is why this adaptation, this is why this Krenz anatomy, to avoid the loss of carbon by photorespiration. That is why the productivity of C4 plants is higher. Fourth question, Rubisco is an enzyme that acts both as a carboxylase and oxygenase. Why do you think Rubisco carries out more carboxylation in C4 plant? Rubisco. In case of C3, what is Rubisco doing? Rubisco is doing carboxylation, that is RUBP accepts carbon dioxide to make phosphoenol pyruvate, that is carboxylation. So, it is helping in carboxylation. During higher temperature, this helps, or this starts working as oxygenase. So, it is also helping in, or it is also taking part in the reaction of oxygenation. So, it is doing two things. Suppose there are 100 Rubisco molecules. Normally carboxylation will take place, but if the conditions are different, half molecules can participate in carboxylation and half can help in oxygenation. In case of C4, C3 cycle takes place in the bundle sheath, and in these bundle sheath cells, carbon dioxide concentration is more. So, Rubisco is acting as only carboxylase. So, this Rubisco helps only in carboxylation. So, if it is helping in only one process, carboxylation will be more as compared to here, because here there is division, some Rubisco in carboxylation and some Rubisco in oxygenation, whereas in C4 cycle, the C4 plants rather, because the site of C3 cycle has been shifted from mesophyll cells to bundle sheath cells, where only carbon dioxide is available and temperature is lower. So, only reaction in which this Rubisco helps is carboxylation. So, our question was, Rubisco is an enzyme that acts both as carboxylase and oxygenase. Why do you think Rubisco carries out more carboxylation? Rather only carboxylation. Rubisco is not there in the mesophyll cells to bind with oxygen and carbon dioxide. Question 5. Suppose there were plants that had a high concentration of chlorophyll B but lacked chlorophyll A. Would it carry out photosynthesis? Then why do plants have chlorophyll B and other accessory pigments? there are parts in this question. Let me read out the first part again. Suppose there are plants that had high concentration of chlorophyll B, but lacked chlorophyll A. That means, suppose there were plants which had plenty of chlorophyll B but no chlorophyll A. Would these plants carry out photosynthesis? So, if there is the question starts with whether, suppose. So, if chlorophyll B is very high and chlorophyll A is absent, will the plant perform photosynthesis? The answer is no. Why is the answer no? And we cannot stop after writing no, this plant will not perform photosynthesis. We have to give justification. Photosynthesis will not take place. Reason? If you remember the photosystem, what is a photosystem made up of? It has many chlorophyll and other accessory pigment molecules, and there is this main chlorophyll which is known as reaction center chlorophyll. This reaction center chlorophyll is always chlorophyll A, and when light falls on all these pigment molecules, they collect these light energy or photons and transfer it to the reaction center chlorophyll. And the electrons which get excited, they come from chlorophyll A. So, the reaction of photosynthesis will start from chlorophyll A. In this condition, they are saying that if there is no chlorophyll A, will photosynthesis take place? The answer is no, because in a photosystem, the reaction center chlorophyll is always chlorophyll A. So, this is the pigment from where the reaction will actually start. So, if there is no chlorophyll A, this reaction will not start. So, first part answer is no. It continues and they say, then why do plants have chlorophyll B? If it is not helping in photosynthesis, then why do they have it? They have it because chlorophyll B, other chlorophylls, carotenoids, xanthophylls, they act as accessory pigments, because chlorophyll B and other pigments act as accessory pigments. What is the role of accessory pigments? Their role is to gather the light energy or photons. So, that is why if chlorophyll A is absent, photosynthesis will not take place. Question number 6. Why is the color of leaf kept in dark frequently yellow or pale green? And which pigment do you think is more stable? So, if why is the color of the leaf kept in dark yellow? So, if you put a plant in dark, they are saying that the leaves turn yellow. The leaves turn yellow. When a plant is exposed to sunlight, in light or in sunlight, the plant synthesizes chlorophyll. So, in light, chlorophyll synthesis takes place. Because if it is in sunlight, it has to perform photosynthesis, and photosynthesis, it requires that chlorophyll pigment. So, if the plant is kept in light, it will synthesize chlorophyll and the leaf would look green. In dark, chlorophyll synthesis will stop, chlorophyll synthesis stops, and chlorophyll gets destroyed. So, chlorophyll is destroyed. If chlorophyll, the green pigment, is destroyed, then what will be the color of the leaf? whichever is the next pigment concentration which is present. So if the next pigment is xanthophyll, which is yellowish brown, so if there are green pigments, that is chlorophyll and xanthophyll. When plant is kept in dark, this chlorophyll is getting destroyed slowly. So now which pigment remains? Xanthophyll, and that is why the color will look first pale and then yellow. So if you put a plant in dark, green color will disappear and the plant will become paler. This is because the chlorophyll synthesis is directly regulated by exposure to sunlight, and if it is in dark, synthesis will stop and chlorophyll will also get destroyed. That means this xanthophyll can survive, or xanthophyll can still remain in dark. So which one is more stable? Xanthophyll. Xanthophyll is, or carotenoids. If not xanthophyll, the other pigment xanthophyll and carotenoids are more stable. So they are stable as compared to chlorophyll. This was question number 6. Let us take the 7th question. 7th question. Look at leaves of the same plant on the shady side and compare it with the leaves on the sunny side, or compare the potted plant kept in sunlight with those in shades. Which of them has leaves that are dark green and why? Basically they want us to compare the color of the leaf. If you are talking about a leaf which is flat, such leaf would have a surface which is exposed to light and a surface which is not exposed to light. So if we are talking about a leaf, this is exposed to light, so this is towards the illuminated side where the light falls, and this is towards the shady side. Which one will have dark green color? Which one would have darker green, and why? Chlorophyll is required for photosynthesis. This surface, the upper surface, because it is getting higher light intensity or more light intensity, it will have some chloroplasts, and these chloroplasts will have some chlorophyll in them. The cells which are on the shady side, these cells, they are not getting enough light falling on them. So they want to capture whatever light is available to them. So in order to do that, they would have more chlorophyll because light is less. So if they have to utilize whatever minimum is available to them, they must have more and more chlorophyll pigment, so that whatever light intensity is available, they are able to absorb everything. So this side will be darker, dark green, and this will be lighter or light green. So illuminated side will be, or exposed side will be light green, and the shady side will be dark green. Reason, the cells of the shady side are receiving less light. To absorb that less light which is available to them, they need more chlorophyll. So they accumulate more and more chlorophyll, and because of which they become dark green. So this lower side will be, or shady side will be, dark green in color. 8th question. 8th question is actually about a figure which is given in the book. The figure is 13.10. Let me draw this figure here quickly. It is actually a graph, and this graph is between the rate of photosynthesis and light intensity. rate of photosynthesis and light intensity. And the graph which they have shown is, it is going up and then it becomes steady. And they have put an arrow here which says A, an arrow here which says B, an arrow here which says C. Then from the end of B, there is a dotted line which is coming down, this is D, and there is one line which is going here, it is E. This is the graph, it is 13.10 and the question is related to this graph. Now let us read the question. Figure 13.10, this is for your reference, it is figure 13.10. Shows the effect of light on the rate of photosynthesis. Based on the graph, answer the following questions. A. At which point A, B or C in the curve is light a limiting factor? In this graph, only light is taken into account. So, all other factors are assumed or presumed to be in plenty. So, at which point A, B or C, A, B or C in the curve is light a limiting factor? We talked about limiting factor. What is a limiting factor? The factor which is available in the least concentration, and that is the one which is going to affect the rate of the reaction. Now, if you see this rate of reaction with increasing light intensity, rate of reaction is decreasing. So, in anywhere in this A segment, when the rate of reaction is not increasing, otherwise it should go up like this. So, it is gradually, slowly going up. So, anywhere in A, we can say that light is the limiting factor. So, anywhere in A. So, A is this entire band. In this band, because light is regulating the rate of photosynthesis, assuming that all other factors are available in sufficient quantity. So, out of this A is the point where anywhere in A we can say that light is limiting factor. B question or B part is, what could be the limiting factors in region A? Here light is one. Can there be anything else also which is acting as a limiting factor? We don't know about other factors, but yes, carbon dioxide can be a limiting factor, water can be a limiting factor, temperature can be a limiting factor, but nothing has been explained in this graph. So, anything that is carbon dioxide, temperature, water, anything can be limiting factor here, unless and until everything is mentioned. We are assuming, we said that light is a limiting factor assuming that everything is in plenty, but suppose the other thing is also less, then that would also become a limiting factor. And the C part is, what do C and D represent on the curve? What is C representing here and what is D representing here? C part, this segment is representing a steady rate of the reaction. So, it represents steady rate of photosynthesis. Here the photosynthesis has come to a steady rate. Here it was going up and then it has become steady. What is D representing? D, if you see this, this is the highest point. After that it has become straight. So it went up and here it reached the highest point. What is that point where the rate of reaction is maximum? We call it optimum. So D represents the optimum light intensity. It is that light intensity where the rate of photosynthesis is maximum, and after that it becomes steady. So D represents optimum light intensity where rate of photosynthesis is maximum. Where photosynthesis is maximum. Let us take the next question. Question 9. Give comparison between the following: C3 and C4 pathways. C3 and C4 pathways. What is meant by C3 pathway? C3 pathway is only Kelvin cycle. And C4 includes fixation of carbon dioxide or acceptance of carbon dioxide by phosphoenol pyruvate. The enzyme which helps is PEPCO. The first stable compound formed is oxaloacetic acid. So all those things. So let us compare this. First comparison, C3 cycle, carbon dioxide, and when we are talking of only C3 cycle, assuming that it takes place in C3 plants, and C4 pathway is what is taking place in C4 plants. So here it is only Kelvin cycle. Here whatever takes place in the mesophyll plus C3 cycle. So it is a combination of both. Carbon dioxide accepted by RuBP. In this case, carbon dioxide accepted by phosphoenol pyruvate, where? In mesophyll cells. In mesophyll. And what happens in the bundle sheet? There also carbon dioxide is accepted. So carbon dioxide accepted by RuBP in bundle sheet cells. The first stable compound that is formed is a 3-carbon compound. So first stable compound is a 3-carbon compound, that is phosphoglyceric acid. In case of C4 plants, the first stable compound is a 4-carbon compound, that is oxaloacetic acid. The enzyme which helps in carboxylation is Rubisco. Here the enzyme which helps in carboxylation are two. One is PEPCO, which is fixing or taking carbon dioxide in the mesophyll cells, and other is Rubisco, which will help in addition of carbon dioxide in bundle sheet. So here it is PEPCO in mesophyll and Rubisco in bundle sheet cells. How many ATP molecules are required to make one glucose with the help of C3 cycle? 18 ATPs for one glucose, molecule. Whereas in case of C4 cycle, it is 2 ATPs more per carbon. So, total 30 ATPs per glucose synthesis. In case of C3 plants which show simple C3 cycle, there is no Kranz anatomy, whereas in case of C4 plants which show the C4 cycle, there is Kranz anatomy. Kranz anatomy is that wreath-like arrangement of big vascular bundle, big bundle sheath cells around the vascular bundle. How many types of chloroplasts are required in C3 plants where only C3 cycle is taking place? Only granule chloroplast. And in case of C4 plants where C4 cycle is taking place, dimorphic chloroplasts are present. Dimorphic, that is granule and agranule. Granule are present in the cells of mesophils and agranule are present in the cells of bundle sheath. So, this is the basic difference between C3 and C4. The second difference which they are, or rather comparison they are asking us, is between cyclic and non-cyclic photophosphorylation. This is part B of 9th question, that is, give comparison between cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic photophosphorylation. In case of cyclic photophosphorylation, only one photosystem participates, and that is only one photosystem, which is photosystem 1. In case of non-cyclic, two photosystems participate. Two photosystems, PS, I am writing 2 first because it is the one with which the reaction starts, PS2 and PS1, both photosystems. In case of cyclic, the electron which is lost from the reaction center, chlorophyll of photosystem 1, comes back to the same chlorophyll. So, electron pair comes back to reaction center chlorophyll of PS 1. Whereas in case of non-cyclic, the electron pair never comes back. It is taken by or finally accepted by NADPH. So, electron pair does not come back to chlorophyll A of PS2. This is the place from where it was released, so it never comes back. How is this electron given back to the reaction center chlorophyll in this case? In case of non-cyclic, this electron is released by photolysis of water. So, in this case, photolysis of water takes place. And in case of cyclic, because the electron is coming back, this photolysis is not there. No photolysis of water. If photolysis is not taking place, will oxygen be released because it is a byproduct of this reaction? So, here no oxygen released. Whereas in case of non-cyclic, oxygen evolves or is released as a byproduct. What is the product? What is that thing that we gain out of cyclic and non-cyclic? In cyclic photophosphorylation, it is only ATP which is synthesized. So, only ATP is obtained. And in case of non-cyclic photophosphorylation, two things are obtained: ATP and the reducing power, that is NADPH. NADPH2 are obtained. What is that condition when these reactions or these pathways take place? Non-cyclic pathway takes place in normal optimum condition. It takes place in optimum conditions. And cyclic takes place when NADPH accumulation takes place. So, this pathway takes place when NADPH2 accumulates. Diagram wise also we know how the difference is. Then we can also write here there is only one electron transport chain. In case of this, there are two electron transport chains, one between PS2 and 1 and second between 1 and NADPH. So, that one more can be a difference, and then we can draw the flow chart also. C part of 9th question. Give comparison between anatomy of leaf in C3 and C4. Anatomy, that means the internal structure they are talking of. In case of C3, if we make the structure of leaf, and in case of C4, if we make the structure of leaf. Upper epidermis, lower epidermis, no significant difference. Lower epidermis will have stomata in both the cases. Upper epidermis has cuticle. In C3 plants, there is xylem and phloem, that is vascular tissue. Here also there is xylem and phloem, which is the vascular tissue. In case of C4 plants, around the vascular tissue there is a wreath-like arrangement of cells. These cells are known as bundle sheath cells. This complete sheath is called bundle sheath. Whereas in case of C3 plants, this differentiation is not there. Here there are parenchymatous cells. Upper few layers are compactly arranged. Lower layers are loosely arranged. So there is no distinct ring of cells around the vascular tissue. All the cells around the vascular tissue are same, and they are all parenchymatous cells. Whereas here all are parenchymatous. Upper end they are compactly arranged, which makes palisade parenchyma, lower half spongy parenchyma, and there are large prominent cells which make a wreath-like arrangement around the vascular tissue. So this is one difference. Difference number 1, that here there is no bundle sheath, whereas in case of C4 plants we see bundle sheath. The second anatomical difference which we find is here only granule chloroplast, and in case of C4 plants there are two types, that is dimorphic chloroplast. One is granule type and the second is agranule. Granule is found in mesophils or mesophyll cells, and agranule is found in bundle sheath cells. So anatomically there are two major differences. One is bundle sheath and second is two types of chloroplast. This arrangement is also known as Cran's anatomy. So we have to mention that no bundle sheath, we can also write there is no Cran's anatomy, and here we find C4 plants show Cran's anatomy, which basically means wreath-like arrangement of bundle sheath cells. So this is the comparison between three things: C3, C4 pathways, then cyclic, non-cyclic, and anatomy of C3 and C4 plants. So we have discussed all the questions which are given after this chapter. Revise the chapter, go through each and every important point, and just keep practicing the questions, and I am sure it is going to be easier after watching all these lectures.